Assalamu alaikum. My name is Hafiz Muhammad Aftab Ayub, and my registration number is 172934. Today's my topic is plant growth regulator. First of all, in plant, many behavior patterns and functions are controlled by hormones. These are chemical influence many patterns of plant development. Plant hormone. What is plant hormone? Plant hormone is a natural substance that are produced by plant that act to control many plant activities. Hormones are produced in one part of the plant and then transported to the other part, where they initiate a response. Then they are stored in a region where stimulus are and then released for transport through the phloems or mesophil when the appropriate stimulus occur. Plant growth regulator include plant hormones, but also include non-nutrients, chemicals not found naturally in plant that when applied to plant influence their growth and development. Now we discuss about classification of hormone. We classify hormone in three different classes. First one is natural hormone, second is synthetic hormone, and exogenous hormones. Natural hormones which are produced by some tissue in the plant, also called endogenous hormones. And synthetic hormones they are produced artificially and similar to the natural hormones in physiological activity, also called exogenous hormones. On the basis of natural function, growth promoting hormone, growth promoter increase the growth of plant. There is also gibberellin cytokinins. In growth inhibiting hormone, there are different hormones which inhibit the growth, such as abscisic acid and thiamine. Also, growth regulator. Many growth regulators are present in plant. There are 5 recognized group of natural plant hormone growth regulators. First one is auxin, second one is gibberellin, third one is cytokinins, fourth one is thiamine, and fifth one is abscisic acid. First of all, we discuss auxins. Auxin influence plant growth. Auxin are found in leaves and stem of the plant. They act as growth regulators and hormones. First function of auxins is cell enlargement or elongations. They are located in meristems and shoot tips. Auxin move mainly from apex of the plant to down. Lengthening of the internode and influence the developing embryo in the seed. Now we discuss about the effect of different auxin in plant growth and development. First of all is cell elongation and cell divisions. Auxin causes growth and elongate the stem due to already existing cell. What is the main functions? What is the main causes of cell elongations? By increasing the osmotic content, permeability of cell to the water and wall synthesis. By reducing wall pressures, we can increase the cell elongations by including the synthesis of RNA and protein, which in turn lead to increase the cell wall plasticity and extensions. Auxins play a important role in apical dominance. Apical or terminal bud of many vascular plants are very active while the lateral bud remain inactive. Removal of apical bud promotes lateral bud to grow. Apical dominance is due to which high auxin content in the apical bud than lateral bud. Other function of auxin is phototropism. Plant bend toward the unilateral light. This is due to high concentration of auxin on the shaded sides. Other function of auxin is geotropism. Movement of a plant organs in response to gravity is known as geotropism. Stem and root accumulated response to gravity. Increased oxygen concentration on the lower side in stem causes those cells to grow move than cell on the upper sides. Stem bend against the force of gravity. On upper side of root grow more rapidly than the lower side and root ultimately grow downward. The other function of auxin is root initiations. Application of auxins to cut and of the stem promote root formations. And the other is control or prevention of abscissions. Auxin abscission does not occur when auxin content is high on distal end and low in proxial end of abscisic zones. Next function of auxin is parathenocarpy. Auxins include parathenocarpy. Parathenocarpy means development of fruit when without fertilizers on seeds. Fertilizations of seeds. Next function of auxin is cellus formations. Undifferentiated mass. What is a cell? Undifferentiated mass of parenchymal tissue is known as cellus. Application of abscisic acid. Application of auxin causes cell to elongate and adventitious roots. Now we discuss about next growth regulator is gibberellin. Gibberellin also called gibberellic acid. Gibberellin has a regulatory many regulatory functions. Many regulatory functions. They are producing shoot effects primarily in the leaf bud and root system. They stimulate stem growth dramatically. Gibberellin stimulate cell divisions, cell elongations, and control enzymes secretions. Example one is in a drought cultivar can be treated with gibberellic acid and grow to normal height. Indicate drought species lack normal leaf of gibberellic acid. Gibberellin involved in overcome the dominancy in seed and buds. Gibberellic acid translocate easily in the plant, able to move freely in both directions because produce is not only shoot effects but also in the root structures. We use gibberellin in the commercial base. They increase fruit size of seedless grapes and stimulate seed germinations and seedling growth. And the next one is now we discuss about cytokinins. Cytokinins promote cell divisions found in all tissue with considerable cell divisions. Example is embryo and germinating seed, young developing fruits. Cytokinins root supply cytokinins upward to the shoot. With auxin to influence differentiation of tissue may be used to stimulate bud formations. As roots begin to grow actively in the spring, they produce large amount of cytokinins that are transported to the shoot, where they cause the development to become active and expand. In tissue culture use cytokinin to induce shoot development. Cytokinin may slow or prevent leaf senescence. And the next one is ethylene. Ethylene is a growth retardant. Ethylene promote ripening, produced in the actively growing meristem of the plant, in ripening or aging fruit, in scaling, in germinating seed, and in certain plant tissue as a response to bending, bonding, or abrasions. Ethylene as a gas diffuse readily throughout the plant. Ethylene is present in gaseous forms Thailand may promote leaf senescence and obsession in leaf fall. Thailand increase female flowers in cucumber will increase fruit productions. The degrading of organs, hormones, and grapefruit, ethylene gas breakdowns, chlorophyll, and then slow downs. And the next one is abscisic acid. Abscisic acid growth retarded induce stomata closing. They also inhibit bud growth and shoot formations. Abscisic acid will spread in a plant body, move readily throughout the plant. Abscisic acid appear to the synthesized by the leaves, interact with other hormones in the plant. As they grow, promoting the effect of auxins. Abscisic acid they promote the effect of auxins and gibberellins. They also involve with leaf and fruit obsessions. Abscisic acid is effective in including a cluster of stomata in leaves, indicating a role in the stress physiology in plant. Example one is that increase in abscisic acid following waters, heat, and high salinity stress.